Okay, so we've gone over the first three of these. This last one, we want to talk about the essential building blocks of life. These are also known as or called macromolecules. So when we're looking at organisms such as what make up the things we eat, we can break down the nutrients that are in there into different types. Okay, so there are 4 different types, and they are called macromolecules. Now, when we're looking at macromolecules, what we're looking at are levels of organization which are larger than the atom, right? You put atoms together and you can make molecules, 2 or more atoms joined together. But those molecules, there are certain ones that can be really, really big. So macro, meaning large, uh, includes these 4 macromolecules, which we find in all living things that are necessary for life. These macromolecules will then make up organelles, which will function together to form cells. And so we're talking about these kind of in between molecule and organelle in levels of organization. All right, so on here, our atoms are the smallest kind of thing that we would study in biology. Molecules are atoms that are all put together in a specific arrangement. And macromolecules, so there's some DNA there which is made up of different types of molecules, different types of structured atoms. Okay, and these are what are important for cellular life because they make organelles which combine to make cells. So what are they? Well, you can find some of these on our nutrition label. They are organic molecules, which means they are carbon-based. And then on those carbons, they have different amounts or different forms or different bonds of hydrogen, nitrogen, and oxygen, and a spattering of other types of atoms and elements in there as well, such as phosphorus and iron and calcium and things like and elements like that. Okay, so our 4 macromolecules of life include carbohydrates, which shows up on the nutrition label, proteins, which we're all familiar with, lipids, which are fats and oils, and the last one, nucleic acid. So this is the one that doesn't show up on the nutrition label, but is very important in the perpetuation of information and reproduction and making new cells and things like that. So we'll spend a little bit more time on nucleic acids, but also go over these other 3. So carbohydrates. These are made of repeating units of carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. But they are more commonly known as sugars. Okay, so carbohydrates are sugars. Our most common one is glucose, which is C6H12O6. There are a few other sugars with the same formula, C6H12O6, meaning there's 6 carbons, 12 hydrogens, and 6 oxygens. So fructose and galactose have that same formula, but they're structured in different ways. Carbohydrates are for energy. That is their main function. They're very simple and easy to break down and use. So we take glucose, and in our mitochondria, we break it down multiple times in lots of different steps, and each time it gives us a usable form of energy called ATP. They can also be for storage of energy. So in plants, you have starch, right? Potatoes are filled with starch. All that is a bunch of glucose molecules chained together and stored in that potato. And then when we eat it, it's very easy to break that down into energy. You and I and other animals will store sugar not in starch, but in a similar molecule called glycogen. And we will store that in our muscles so that when we exercise, we can use it readily. Some carbohydrates are structural, meaning they're important for the support of an organism. Chitin is one found in shells of arthropods. It's also found in fungi. And then cellulose and lignin. These are the undigestible parts of plants which form the cell walls and the parts that make plants rigid and tough and hard. So those are also made out of carbohydrates, and they're not for energy mostly, but for the structural component of those organisms. All right, proteins. Proteins are the functional units of the cell. So this is what's kind of doing all the work in different cells or in multicellular organisms. And they have a lot of different functions. They can be enzymes. Enzymes are going to help reactions go at a faster rate. Hormones, right? So they are for signaling molecules that will, when attached to the right cell with the right receptor, cause a change in that cell. They are structural, so they form parts of muscles and tendons and skin that hold things together, make things stick, make things stiff and rigid, and so on and so forth. They're also important for the immune system, so some of them will be able to attach and recognize different microorganisms that are pathogenic or would make you sick. Transport, so some of them will take something needed from one cell and transport it to another cell, or they will help things pass from one part of the cell to another, or inside and outside of the cell. Okay, lipids are mostly fats and oils. Waxes also are lipids. These are hydrophobic in nature. So by definition, they do not mix with water. That's what hydrophobic means. Okay. Some of them have some functional things. So oil on feathers of birds help to make that repel water and makes them waterproof. They can be for insulation, such as in this whale that is being carved up. That blubber layer is to protect it from the cold environment in which it lives. They can be for energy storage, so they can have dual function as well. So this whale has a thick layer of blubber, but that blubber can also be used for energy if needed, and broken down and used in cells to acquire energy. And some of them are structural. So every one of our cells has a lipid bilayer, and that's what this bottom right figure is showing. Those tails kind of look like bobby pins, not the head, but the tails are fatty acids, are lipids that form this semi-permeable membrane around each and every one of our cells. And they can also be hormonal, so your steroid hormones such as progesterone, estrogen, testosterone, those are all hormones which signal changes in the body throughout your life and developmental periods. Okay, so we've kind of established the different macromolecules. We can talk about, all right, well, there's the buns, which are made of carbohydrates, the cheese, which is made of fats, the burger, which is made of protein, the lettuce, which is made of carbohydrates, including structural carbohydrates such as cellulose. All of these macromolecules you can see in here. The one that's harder to see, however, is nucleic acids, because it's actually made of sugars and other things, and isn't a component that is a large component of the things that we digest when we eat. So this one is not found on our nutrition label. Nucleic acids are made of nucleotides. They include DNA and RNA, deoxyribonucleic acid and ribonucleic acid. So that refers to the type of sugar that is used in those nucleotides. RNA has ribose, is the sugar it used, so it's ribonucleic acid. And DNA has deoxyribose, this is the sugar that it's used, so it's deoxyribonucleic acid. DNA provides the instructions for making proteins, and RNA helps make the proteins. And the way that we get from DNA to proteins is through RNA, and we'll talk about that in a minute. There are different types of RNA. Messenger RNA is like a template that is read. Transfer RNA reads the messenger RNA and brings amino acids. And ribosomal RNA makes the ribosome, which helps string those amino acids together. All right, so the structure of DNA, again, it is a nucleic acid. It is made of nucleotides. And this is probably, we aren't going to go into this too much, but essentially you see on the bottom here, 3 phosphate groups. That's a P surrounded by 4 O's. Each one of those is a phosphate. Okay, you have 1 sugar, that's the yellow part there, and it's a 5-carbon sugar, pentose. And it's either deoxyribose or ribose, and then a base in blue. And it doesn't really give you much about the base, but there are 5 different types of bases used in these nucleic acids, and they're often just abbreviated by A, G, T, C, or U. All right, and there's a specific way that they attach to each other, these nucleotides. All right, and the way that they attach is through what's called a phosphodiester bond. So I've circled the phosphodiester bonds here. You have that yellow nucleotide on the left attached to the blue nucleotide on the left, and the circled in red, the phosphodiester bond between them. And so you can make a very, very long chain of these by just making more and more phosphodiester bonds. On the other side, you have nucleotides that are flipped upside down in orientation to the other ones. And so they are the DNA, which has 2 strands of nucleotides, all tied together through phosphodiester bonds, are then in opposite direction toward each other, and then they make bonds in between. So those dotted lines are the hydrogen bonds which form in between. It's a kind of it's a complex structure, but once you understand the basics, you can see how it holds information that can be used. All right, so one of the things that was discovered was that the number of adenines is always equal to the number of thymines, and guanines is always the same as the number of cytosines. And the reason that is because these ones, when they are across from each other on DNA, they will form those hydrogen bonds between them. So A will always form bonds between T, and G will always form between C. And this is probably something you remember from high school biology. All right, Rosalind Franklin was a scientist who discovered the shape of DNA, that it forms a helix, meaning it's like a ladder that's twisted. And she even discovered the actual amount, and that's based on these images she made using X-ray diffraction. Now, when I look at this image, I do not see what she sees, but somehow this image means that you have a double helix, and it's a twisting ladder, essentially. And so she did a lot of science in order to get to that point. And then Watson and Crick were able to take her work and other people's work and put together a full 3D model of what DNA looks like. Okay, and so I guess I just said this, but Watson and Crick did not do any experiments. They took other people's work and they kind of put all the pieces together and were able to make a model of what DNA looks like. Oh, I don't have a picture of the model, but basically it is this double helix that we talked about before. And they are credited for the discovery of the shape, even though they didn't do any experiments for it. Okay, so here's the shape, 2 strands of nucleotides in opposite direction, facing each other, with phosphodiester bonds connecting those nucleotides along the ladder, forming the sides, and then the rungs of the ladder are formed by those hydrogen bonds, those dotted lines. And G's and C's form 3 hydrogen bonds. A's and T's form 2 hydrogen bonds. All right, so we've talked about our nucleotides, DNA and RNA, and then the functional products of the cell are proteins. So DNA has the instructions for how to make RNA, okay? And so we use DNA, it's read in a process called transcription, and then RNA is made from that template. And so we have this double helix, this 2 strands of nucleotides being converted into 1 strand of nucleotide in RNA. And then that RNA is shipped to a ribosome, and that ribosome will read the RNA, and based on the code of the order of A, G, C's, and U's, it will make specific proteins, and that process is called translation. So DNA and RNA are in the same language. They are both nucleotides. So a transcription is something that where you would use the same language and just write it in a different medium. Whereas a translation is going from one language to another. RNA is a nucleotide, or it's a nucleic acid. Proteins is a different macromolecule altogether. It's made of amino acids, so that is a translation process. So again, DNA uses 4 bases, A, G, C, and T. RNA uses 4 bases, but it uses a uracil instead of a thymine. Again, is the similar language and is a single strand rather than a double strand. And then proteins language is in amino acids. There's 20 different types of amino acids. They all have different properties, which are important because when you string them together in different ways, then you can form different functional products. You can make them strong or flexible or soluble or whatever the function is. You can just mix up these 20 different amino acids in order to do that. And then the order of those amino acids is determined by the RNA as it's read, which is determined by the codes which are in the DNA. And that then forms all the different things you need to survive and live in your proteins. Okay, so a protein then has this very complex interfolded interactions of amino acids, and 3D models look like these big squibbly things, but that's what they are.